chapter we'll be talking about experiments. Concepts. We'll distinguish between observation and experiment. We'll define subjects, factors, and treatments. Again, we'll show how to experiment badly. Talk about randomized comparative experiments, cautions about experimentation, and block designs. So in contrast to observational studies, experiments don't just observe individuals or ask them questions. They actively impose some treatment in order to measure the response. So an observational study observes the individuals and measures variables of interest, but does not attempt to influence the responses. The purpose is to describe some group or situation. Whereas an experiment deliberately imposes some treatment on individuals to measure their responses. The purpose is to study whether the treatment causes a change in the response. When our goal is to understand cause and effect, experiments are the only source of fully convincing data. The distinction between observational study and experiment is one of the most important in statistics. Observational studies of the effect of one variable on another often fail because of confounding between the explanatory variable and one or more lurking variables. A lurking variable is a variable that is not among the explanatory or response variables in a study, but that may influence the response variable. Confounding occurs when two variables are associated in such a way that their effects on a response variable cannot be distinguished from each other. Well-designed experiments take steps to avoid a few examples. We had this earlier. Ice cream consumption and drowning deaths are very highly correlated. We might try to conclude that eating ice cream causes people to drown. But it's a lot more likely that there's a third variable, namely the time of year, that influences both these variables. So that third variable is the confounding variable. Another example. Suppose a research group designs a study to determine if heavy drinkers die at a younger age. Their results show that people who drink excessively are likely to die younger. But it's quite possible that the heaviest drinkers came from a different background or social group. Heavy drinkers may also be more likely to smoke or eat junk food, all of which could be factors in reducing longevity. A third variable may have adversely influenced the results. Okay, now we'll define a few things here. An experiment is a statistical study in which we actually do something. This is a treatment to people, animals, or objects, the experimental units, to observe the response. Here's the basic vocabulary. The experimental units are the smallest collection of individuals to which treatments are applied. When the units are human beings, they're often called subjects. Animals are often called subjects as well. The explanatory variables in an experiment are often called factors. A specific condition applied to the individuals in an experiment is called a treatment. If an experiment has several explanatory variables, a treatment is a combination of specific values of these variables. How to experiment badly? Experiments are the preferred method for examining the effect of one variable on another. By imposing the specific treatment of interest and controlling other influences, we can pin down cause and effect. Good designs are essential for effective experiments, just as they are for sampling. So here's an example. A high school regularly offers a review course to prepare students for the SAT. This year, budget cuts will allow the school to offer only an online version of the course. And so we have the subjects are the students, the treatment is the online course, and the response is SAT scores. So over the past 10 years, the average SAT score of students in the classroom course was 1620. The online group gets an average score of 1780. That's roughly 10% higher than the long-time average for those who took the classroom review course. So, is the online course more effective? Well, we can't really say because we have nothing to compare it to. So, yes, it was higher. There may be compounding variables and lurking variables. We don't know. There's nothing with which to compare it. Many laboratory experiments use a design like the one in the online SAT course example. We take experimental units, we apply a treatment, and we measure the response. In a laboratory environment, these designs often work well. Field experiments and experiments with animals or people deal with much more variable conditions. Outside the laboratory, badly designed experiments often yield worthless results because of confounding. Look at it. So the remedy for this is to perform what's called a comparative experiment, in which some units receive one treatment and similar units receive another. The most well-designed experiments compare two or more treatments. Comparison alone isn't enough, though. If the treatments are given to groups that differ greatly, bias will result. The solution to the problem of bias is random assignment. So, in an experiment, random assignment means that experimental units are assigned to treatments at random, that is, using some sort of chance process. In a completely randomized design, the treatments are assigned to all the experimental units completely by chance. Some experiments may include what's called a control group that receives an inactive treatment, like a placebo, or some existing baseline treatment, like if we're measuring, testing new types of curricula for teaching math or something, the baseline might be the standard approach. So we take the experimental units, we randomly assign the groups, the people or the subjects in this case, into two groups. We apply the two treatments, we get responses, and then we compare the results. The logic of randomized comparative designs. These are designed to give good evidence the difference in the treatment actually caused the differences we see in the response. So essentially, everything else being equal, the only difference is the treatment that gets applied. So we control for lurking variables that might affect the response, most simply by comparing two or more treatments. We randomize, use chance to assign the experimental units to treatments. And replication. We use enough experimental units, i.e. the sample size, in each group to reduce chance variation in the results. Once again, the larger the samples, the less variation we're going to have. An observed effect so large that it would rarely occur by chance is called statistically significant. A statistically significant association in data from a well-designed experiment does imply causation. Some of the cautions. The logic of the randomized comparative experiment depends on our ability to treat all subjects the same in every way except for the actual treatments being compared. In a double-blind experiment, neither the subjects nor those who interact with them and measure the response variable know which treatment a subject receives. Now, there's another type of assignment called block designs. There are some times when the simplest method just does not yield the most precise results. So, for example, if we know that there are differences between, say, men and women, or old and young, or something like that, in the way that they're going to respond to a particular treatment, then by not separating those units out and looking at them separately, we run the risk of not getting an actual statistically significant value, even when there is one. So, a block is a group of experimental units that are known before the experiment to be similar in some way that is expected to affect the response to the treatments. In a block design, the random assignment of experimental units to treatments is carried out separately within each block. So, we form blocks based on the most important unavoidable sources of variability. So, if we know what the lurking variables are, randomization will average out the effect of the remaining lurking variables and allow an unbiased comparison of the treatments. So, we control what we can, we block on what you can't control, and then randomize to create comparable groups. So here is a sort of a design of an experiment. So we begin with a group of subjects. And each of these subjects are going to look at 3 different advertisements. And they're going to have their reactions compared. So how do these subjects respond to 3 different advertisements? Well, we may know up front that women and men will automatically respond differently to different types of advertisements. We know that, so that is a lurking variable. We recognize that up front, so we use blocking to separate those out. So we immediately break the subjects into groups of women and men. This is not random. But then, at that point, we then randomly assign all the women to one of 3 groups, and we randomly assign all the men to one of 3 groups. So we have a total of 6. And then each gets exposure to the advertisement, and we compare the reaction. Now, we can't necessarily compare men and women directly, but again, we know that there are going to be differences. So we compare women's reactions to the 3 ads and men's reactions to the 3 ads. So to review, we talked a little bit about observations and experiments and the distinctions between them. We identify subjects, factors, and treatments. How to experiment badly. Designing randomized comparative experiments. Some of the cautions about experimentation. And block designs.